the last presenter for this morning, we have Ludovic Humbert, which is an expert in computational imaging. His background is in biomedical and biomechanical engineering. And so he is the founder, co-founder of Galgo Medicals and the founder of 3D Shaper. So he's really giving an insight of how academic and industrial collaborations look like. He's also one of the principal investigators in our Disk for All consortium and other European-founded project. So yeah, today we're talking about, one of his area of expertise, which is the shape and the modeling from 2D images to 3D models, especially in osteoporosis today. So.
Okay. Thank you, Paola, for the kind introduction, and good morning, everybody. So yeah, today I will basically focus on the 3D modeling methods that we developed in my company, 3D Shaper Medical. So I'll start with a bit of some words on our history, which actually started here at UPF some years ago. So we were some researchers and software engineers working in the lab in medical images, and we decided to create a company, Galgo Medical. So it was there 10 years ago, and you see our face over there in that photo. So yeah, we were 7 co-founders at that time. The company is based in Barcelona, and we basically make software in medical images in those 3 areas. So in cardiology for the treatment of arrhythmia, in musculoskeletal imaging for the treatment of osteoporosis, which is my area, and in angiology for the treatment of aneurysm. Okay, then later on we incorporated a 4th line in brain imaging for the treatment of epilepsy. And then 3 years ago, we started to create dedicated companies for each of the lines that we had at Galgo Medical. So we started with the line in cardiology, and we created that company, ADAS 3D Medical, which is actually a joint venture of Galgo Medical and Circle, which is a company based in Canada. And then we replicated a very similar scheme for my activity. So we created in 2020, in the middle of the pandemic, 3D Shaper Medical. And then here we went through a process of fundraising that we closed in June 2021. So the company has grown a lot. So we now 40 people as a total, if we include people at Galgo Medical, ADAS 3D Medical, and 3D Shaper Medical. And in my company, we've always been like 5 to 6 people working on bone imaging, and now we are a team of 11 people. Okay, so today I will focus on our activity at 3D Shaper Medical, which is also a company here based in Barcelona, and here the focus is musculoskeletal diseases. So this is our team, so those 11 people I was mentioning you, so we have more people oriented towards the research and other people more focused on the development of the product. And we also have 2 open positions, okay, over there. One for a software developer, another one for a scientist in medical images. So there's a lot of people here in the room today. So if anybody is interested or anybody knows someone who would be interested, please just get in touch with me, okay? So the pathology that we address at 3D Shaper is osteoporosis. Okay, so osteoporosis is characterized by a low bone density. So that's a pathology that actually affects more than 200 million people worldwide. Okay, and the issue is these low bone densities that the patient they are more likely to fracture. So we have 9 million fractures each year, which is a fracture, an osteoporotic fracture every 3 seconds. And yeah, again, it's a pathology that affects more women than men. So I'm saying again because Keita just just discuss about the osteoporosis, scoliosis that was also affecting more girls than boys. So here it's the same. It's more for elderly people, but yeah, we'll have 1 of 3 women over 50 that will experience an osteoporotic fracture, and 1 of 3 men. Okay, and in all the fracture that you can, so you can fracture your wrist, you can fracture your ankle, but the most serious fracture that you can get are the one at the hip, which are actually 18% of the osteoporotic fracture, and the one at the spine, 15%. So the pathology is associated also with a high cost for healthcare system. So the cost in Europe in 2019 was up to 57 billion. And here, interestingly, in spite of the fact that the hip fracture is only 18% of the total fracture, it's actually more than half of the cost for the treatment of osteoporotic fracture that are related to the hip. So then, because of the population aging, it is something that is going to increase. Okay, so the forecast is that this is likely to increase by 25% within 10 years. So the number of osteoporotic fracture, and this is also something that is associated with an increased mortality in this elderly population. So we actually have more than 20% of the patients who will die within 1 year after hip fracture, and only 20% of the patients will completely recover after a hip fracture. Okay, so now in clinical practice, the physicians, what they use to diagnose osteoporosis is DEXA, so dual energy X-ray absorptiometry, which is a device that is very similar to a conventional X-ray device. Okay, but instead of taking only 1 image, it takes 2 times the same image with a different energy. Okay, so it takes 1 low energy image and 1 high energy image, and then the system will combine those 2 images to create an image where you would see only the bone. Okay, which is an image like that. So here you would see the femur and the pelvis, and that's also a quantitative image. So, which means that here at every pixel, what you measure is the projected bone density. Okay, so you can quantify, you can measure the bone density in that image. So here with that image, typically what the physician would do is that the system would calculate the bone density in specific area. So the area that is used the most is the total femur, which includes the neck, here the trochanter and the shaft. So the system would measure give you 1 value, which is the bone density, the average bone density in this area, and then it would use age-related reference data, such as this graph, to compare that value of this patient to the young normal population. Okay, so if the point, so here you have the normal bone density in green, then you have here osteopenia, which is a low bone density, and here you have the osteoporosis range, which is the pathology. So in this graph, what we also have is the average density here in the general population against the age. So here what you see is that so that's the curve for women. We're all losing bone density. So there is something that's happening to everybody. It's not a pathology, it's a natural process. And here in the case of the women, the loss gets faster after between 40 to 60 years old because of menopause. Okay, and this is also the reason why women they get more fracture compared to men. So that's basically what the physicians does in daily clinical practice to diagnose osteoporosis. So the good thing with DEXA is that the device is very cheap, and the exam is very cheap, it's easy. Also, the radiation dose is very low, so you can screen the population with the device, and then you can monitor your patient, so you can scan the patient every year. There's no problem in terms of radiation dose. But there's one big limitation of DEXA, which is the fact that you get, that's a 2D analysis. Okay, so you get only one measurement of the 2D bone density of the patient, and you don't take into account the 3D distribution of the bone density. You don't take into account either the cortical and trabecular bone density, so you cannot have a separate measurement of those two compartments. So cortical bone, if you look at a cross-section here of the femur, so you have this dense layer, which is the outer bone, which is the cortical bone here. And then in the inside, you have this spongy bone, which is the trabecular bone. So both compartments are important for the strength of the bone. And with DEXA, you don't have access to that information. Okay, so then if you would like to have this information in daily clinical practice, you could use CT, right? Because CT, you can have a 3D analysis. You could measure the cortical bone. You could measure the trabecular bone. Okay, so you would use CT, and then you would also include those. So from a CT, you would convert it into a QCT, quantitative CT, by using those kind of phantoms. Okay, that would. So this is a phantom that you scan together with the patient, which is here under the table. And then you see the phantom here in the image. So the phantom has some tubes with known value of bone density, and then you would use those values. You will measure those values to calibrate the image, and then to be able to measure the bone density in the image. Okay, so there is a solution here. The only thing is that the device is much more expensive compared to a DEXA scanner. The exam is also more expensive, and I would say that here the main issue is the radiation dose. Okay, maybe you could scan your patient once to make a diagnosis, but then you couldn't monitor your patient and have him getting a CT scan every year to monitor the progress of the disease. So you couldn't do that. So because of this, this device is actually not used in clinical practice for the management of osteoporosis. Okay. So here comes our technology, 3D Shaper, our software, where here the goal is to kind of overcome the limitation of both modality, CT and DEXA. So what we use here is DEXA because it's the system that is used for the management of osteoporosis. And what our software does is that it models the bone in 3D using this 2D DEXA image. Okay. So how do we do it? We use a statistical model of the bone shape and bone density here that we build from a database of CT images of a population. Okay, so we use those old school shape and appearance model that we build using principal component analysis, and then what you get are the main modes of variation of both the shape and the density that you see in that image. Okay, so here you have the first mode, the second mode, the third mode, and you have a series of modes that represent your statistical model. And then when you have the DEXA scan of the patient, okay, over there at the right, what we are going to do is that we are going to register this model in a 3D to 2D registration process, which is automated. So what you are going to do is that we're going to change the pose of the model, we're going to change the size, and we're going to change the shape and the density according to the modes of our statistical model, so that the projection of the model that you can see in this area, okay, so that's here, that's the projection of the model, is the same as the DEXA scan that you have here in that image. Okay, so I will show it to you here in this video. So the model is adjusting, change the rotation, and then we're going to change the shape and the density, and you see that here the projection, it gets closer to what's the DEXA scan that is behind. Okay, so that's a fully automated process, and then here at the end of this process, what we get is a subject-specific QCT-like model of your patient, but from its DEXA scan. So then when we have this, we've developed a method to automatically segment the cortical layer. Okay, so here we've developed the method where we can provide an accurate segmentation that kind of handle the partial volume effect that you have in your image. So we just, we fit a mathematical function of the cortical thickness, the density, its location, onto onto density profiles, okay, that we measure in the image. And at the end of the process, what we get is a segmentation of both the cortical and also the trabecular compartments. So with this segmentation, we are able to calculate a lot of measurements in both compartments of the bone. So we've integrated those algorithms in a software, okay, the 3D Shaper is the name of the software, that can be used in clinical practice, okay, so for the diagnosis of osteoporosis. And here in clinical practice, we only output two measurements, okay, so we output the cortical density and the trabecular density at this region of interest, okay. So the software is compatible with most of DEXA manufacturers here, and it's registered as a Class 2A medical device. Okay, so we are registered in Europe and also in those countries where we sell our product, and we've submitted to the FDA 2 months ago our first 510K dossier, and this is under evaluation. So this would be to be able to sell the product in the US. So that's for clinical practice, and then we have another version of our software which is more for clinical research. Okay, so here we output much more measurements. We actually have 72 bone parameters that we calculate in those regions of interest. So at the neck, at the trochanter, at the shaft, we calculate the cortical thickness, its density. We have those biomechanical measurements also, this moment of inertia, section modulus, this buckling ratio. And with that version of the software, one good thing is that we can retrospectively analyze data that the clinician might have already in the context of a clinical trial, for example. So imagine that the clinician would have like 2,000 DEXA images in a clinical trial. What you would do is just to import all the images in our software. So the software will process all the images and then output a spreadsheet with all the measurements for every DEXA scans for him to do the statistics. Okay, so it's pretty automated and pretty easy to use for the clinician. So then from a validation perspective, the first thing that we done here is to validate the accuracy of our measurements. Okay, so here we perform a study where we compared 3D Shaper analysis to QCT, which is the gold standard here. We have a population of, we've included in that study 157 subjects, men and women, and patients with different bone conditions in terms of bone density. Here you see 3 examples in this from those 157 patients. So where we compare so here that's the cortical thickness in these color-coded images. So you see that the anatomical distribution of the cortical thickness is quite similar if you compare 3D Shaper to QCT. Okay, in those 3 cases. And here at the right, what you see is a cross-section. So it's the frontal cross-section that we generate with 3D Shaper and that you can get also with QCT. So you see also that they're quite similar, and it's interesting to see that your patient 2 has a very thin cortex over there. But this patient has a thicker cortex, and you see it in both with both modalities. And then that's the more quantitative evaluation of this. So if we take the two measurements that we use that are used in clinical practice, which is the cortical density and the trabecular density, so this is the correlation that we have between our measurements and the same measurements but computed using QCT. So the measurements are quite accurate compared to that gold standard. Okay, so we have the software 3D Shaper has been used in quite a lot of clinical studies, and we have also an interesting number of journal publications and presentation conferences using it. We have clinical studies with pharma companies based in the US. We also have last year an interesting work with NASA, where they use our software to process the data from astronauts. We have collaboration with scientific society in osteoporosis and key opinion leaders using our software. And here, in terms of clinical research, the two areas that are investigated the most, we have two areas that are investigating the most with our software. And the first one is the association, how our measurements are associated with osteoporotic fracture. So that's just an overview of the studies that look into this. And then here the main results that we found from those studies, that this measurement at the trabecular bone, so the trabecular density, was found to significantly improve the predictive ability of hip fracture compared to DEXA. So say here is that we could potentially improve the fracture risk prediction using 3D Shaper compared to the use of DEXA alone. And then the second area where our software has been used a lot is to monitor the effect of treatment. Okay, so osteoporosis treatment. So those are the studies that has been performed on that topic. And here the little circles that you see are the different drugs that were investigated using our software. Okay, and here I would give you an example of this work that we've done with Radius, which is a pharma company based in the US. So Radius, they manufacture this drug, this one, which is abaloparatide. Okay, and they have this trial, this active trial, when they included patients that were randomized at baseline in 3 groups. Okay, so they have a group receiving placebo. Okay, another group receiving abaloparatide, and another group receiving teriparatide, which is another drug manufactured by Lilly. Okay, so then the patient, they had the DEXA scan at baseline, at 6 months, and at 18 months. Okay, so when what they found with DEXA, okay, so here no 3D Shaper, with DEXA, is that both drugs, both abaloparatide and teriparatide, they found a marked increase in bone density with both drugs, but the magnitude of the increase was pretty similar with the 2 drugs. Okay, so then when we've analyzed those data with 3D Shaper, what we actually find is that the increase in cortical thickness over there and the increase in trabecular density here was also very similar with the 2 drugs, but actually the differentiation between the 2 drugs was actually in the cortical bone and was with the cortical density. Okay, so here you see here that abaloparatide has a more marked resulted in more marked increase in cortical density compared to here to teriparatide. Okay, so and if you compare the difference here compared to DEXA, you see that the difference is much more higher. Okay, so here we were able to show that so this is the anatomical distribution of the changes in cortical density after 18 months of abaloparatide, and here you can compare with the distribution of the changes under teriparatide. Okay. And then we were also able to generate cross sections that shows the changes here at month 18 with the 2 drugs, the 2 drug treatments. So here what you see is just it's a frontal cross section. So here you'll be seeing both the changes in cortical density and trabecular density. So everything which is in blue-green means an increase in bone density, and everything that is in yellow red means a decrease in bone density. So here, frontal cross-section, and then we also generated those 3 cross-sections at the neck, the trochanter, and the shaft that you see here. And here what's interesting to see is that with teriparatide at month 18, you still have those like not completely mineralized bone area that you can see here in yellow, which are actually in the middle of the cortical bone, that you don't see with abaloparatide. Okay, and we also created those 3D images. Okay, where here what we've done is that we just removed all the region of interest where the bone was what the bone density was increasing, to just keep the region of interest where the bone density was decreasing. Okay, so abaloparatide, you see there is only this region remaining, and teriparatide, you still see a lot of regions that are not completely mineralized. So I have to go over there to just run that's a video. I have to go over there to run it, I think. The map. So this one is teriparatide. So that's the yellow area which are in the middle of the cortical bone, and here with abaloparatide. Okay, so then as I was telling you, so there's a lot of drug treatment that have been processed with 3D Shaper within some clinical studies. We've actually processed all those treatments. So now we're able to create a kind of a table where we know what's the effect of the treatment on both compartments. Okay, so that includes the trabecular density, here the cortical surface density, and then we have also the thickness and the density. But if you remember, in clinical practice, they would just use those two measurements, okay? And so that's the pattern of each drug analyzed by 3D Shaper. So here what's interesting is to see that you can have a treatment that, for example, if you compare alendronate to teriparatide. Okay? So if you just use DEXA to analyze the effect of both treatment, you would see a very similar increase in bone density. While actually what's happening inside the bone is completely different, because you would have teriparatide would increase a lot your trabecular density, but you have no effect on the cortical bone. While with alendronate, you know, it's like the increase is shared between the trabecular and the cortical compartment. So then, how do we take that to the clinics? Here, the idea is very simple: is just to be able to detect if a patient, for example, has an imbalance between the trabecular and the cortical density, that to be able to detect it with 3D Shaper. Okay, so that's the case. That's not a real patient; it's just. But to be able to detect that, for example, here, this patient has a trabecular bone in the red area, a cortical bone in the yellow area. So here, the idea for the physician will be to give that patient a drug that would increase the trabecular density. Okay, but then he wouldn't really care on the effect on the cortical density. And then this way, after 2 years of treatment, maybe hopefully you would have taken the trabecular density into the safe zone. Okay? So that's a bit the idea of how 3D Shaper can be used in clinical practice. So here we have an actual clinical case of a woman, 68 years old. For this woman, the decision was made to treat her with teriparatide. And then this was the 3D Shaper analysis at baseline. Okay, so here the trabecular bone is in the red area, the cortical bone in the yellow area. So the clinician gave teriparatide, which is a treatment that's actually increasing the trabecular density. So it was a good decision for that patient. And then after 18 months of treatment, the trabecular density was taken to the yellow area. So it's a bit borderline yet, but it's improving, no? And then the cortical bone, there's no effect, but you wouldn't really care because the bone is here in the middle of this yellow area. So here it's good is that it would help the clinician to choose what's the best treatment for the patient, and then also after giving the patient, you would be able to validate that the treatment was actually the appropriate treatment for that for my particular patient. Okay, then we also have some ongoing developments. We have a very similar method at the spine. Okay, so we have a statistical model of the lumbar spine, L1 to L4. shape and density, and then here we take the DEXA scan of the patient, and we register the model onto the DEXA scan of the patient to get, again, a CT-like model of the patient. It has been validated against QCT, and we have clinical studies ongoing there at the spine. And then we have another method that we are actually developing with the UPF, with Jérôme's group, which allow us to run a finite element analysis to actually measure the strength of the femur. So here the idea is we take the output of 3D Shaper, okay, which is basically a surface mesh and the bone density image. We create a 3D mesh, and then we run a simulation of a sideways fold to measure the strength of the femur. Okay, so we have now a fully automated pipeline that we validated against QCT. Okay, so that's and we get quite nice correlation between the 3D Shaper-based strength measurement and QCT-based measurements. And we've also been testing this method to capture the changes in bone strength for patients under pharmacological treatments. And then what we get is also very consistent to what's expected. So with this method, we can compute the overall strength of the femur, but also the strength of the trabecular bone and the strength of the cortical bone. And here what we get is what is expected. With teriparatide, we have this increase in trabecular strength, but no change in the cortical strength compared to denosumab, where both compartments will increase in strength. And this is also what we see in those images. Okay, so the stress, you see it's all blue for denosumab, where when you look at teriparatide, it's blue in the inside because of the increase in trabecular strength, but it's yellow at the outside because of this no change in cortical strength. So I think that's what I wanted to show you today, and thank you very much for your attention, and I would be happy to take questions.